Can everybody see that? Could I get a thumbs up if you can see that? Yay, cool, awesome. All right, so everybody, welcome to Project Week. We are so excited to have you here. As you see, we are kind of standing up on the stage right now in front of the podium. So when it's your time to present your project plan pitches, come up here and stand on the podiums. That way you can broadcast to the whole room and everyone can see you and hear you and see your screen when you share it. So I just wanted to send a welcome to everyone to start things off. You made it to the GatherTown, yay. This is gonna be your virtual space for Project Week. You can also feel free to use a Jitsi room or a personal Zoom room to touch base with your team. Obviously, since you're here, you know the password to GatherTown, which is ABCD-ReproNim course, oops. But if any of your team members are having any problems, that's the password to join. We do recommend using Google Chrome for GatherTown. Safari's kind of still in a beta stage. And if you have any issues joining the GatherTown, please post to the Slack so that we can help troubleshoot. And then also, if you haven't explored already, explore the space a little bit. There's a game room with a lot of different games. You can kind of ghost through objects or people if you need to get past them by pressing G, and you can also do a little dance by pressing and holding down Z. For Project Week, we recommend that you check out our Project Week outline, Project Week fact. We'll be updating that periodically with schedules as they come out. Particularly, we've invited some of the ABCD-ReproNim instructors to join us throughout the week, so we'll be updating the Project Week outline with those schedules as they come. All right, so I just want to, we've seen a bit of chatter on the Slack so far in the various project teams, and I am so happy to see that. I just wanted to give a little, I don't know, guidelines on who your helpers are this week. So primarily, your helpers for your projects for this week are your team members. So if you haven't already, join a team. All the Slack channels that we've set up for each project are actually private because we only want people who have ducks to be on those Project Week specific Slack channels. So if you haven't joined a team already, do it and then ask us to add you to the specific Slack channel. Also, all the other people in Project Week are your resources this week. So if you have a question about something, post it to the Project Week Slack channel. And if you have an answer, great. Try and answer questions that you know answers to and post questions that you don't know answers to. So that'll be our primary place of communication. Also, all the ABCD-ReproNim organizers, Angie Laird, Dave Kennedy, Satra Ghosh, and JB Pauline, and myself will be hanging around the Slack and the GatherTown to be able to help any team that needs help with the Jupyter Hub or anything that comes up. We're also going to have some select ReproNim instructors kind of just hanging around the Slack and being available on a rolling basis to answer any ReproNim related questions. So again, post any questions that you have to the Project Week channel and we'll get some people to help answer those. For this week, TAs are primarily going to work kind of as conduits to direct your questions to the appropriate places. Although if a TA has the time and the interest, they may actually end up joining your team as a team member. So if that happens, they're kind of taking off their TA hat and putting on just a regular team member contributor hat. So if that's the case, just treat them as you would normally treat a team member. And if you have any questions, either direct them to one of the organizers or to the Project Week Slack channel or to a TA who will then pass the question on to somebody else probably. All right, so for today, the schedule for today is we just started our kind of welcome here to Project Week, but after this, we're going to jump into the project plan presentations. And that's where teams are going to kind of talk about what the focus is that they plan to work on for the week, what the kind of action plan is to try and make it happen, and what are their midweek and end of week goals. So if you haven't already picked a project team, I know some of you have because we asked you to do some work in this past interval week, but if you haven't already picked a project team to work with, that is okay. Just pay attention to these project presentations and then see which one you want to work on, and then message them and message us so we can add you to their Slack channel. And then after those, at around 2 p.m. Eastern Standard Time, we'd like you to go and meet up with your team to start working on the projects that you've outlined. So part of that task for today would be make sure that you're on your project Slack channel, set up a GitHub page for the project if you guys haven't already, make sure you all have access to the Jupyter Hub. And really importantly, one thing that we'd like you to do today is decide on a daily time to check in with your team for the week. We're going to be asking you to check in with your team once each day to just go over a project. And then for the rest of the day, plan what you want to work on and then just get started. And with all of these schedules things, we've ended them at 5 p.m. Eastern Standard Time, but feel free to keep working until your normal end of the workday. I know we have a lot of different time zones represented here. The most important part is that we just want you to check in with your team once a day. All right, so then on Tuesday through Thursday, that's going to be the team project hacking portion of this week. That's where most of your work is going to get done. So whenever your normal working day is when we'd love to see you working on your various projects. So our guidelines here is we'd love to see each team start the day with a clear plan on what your team members will work on and what specific goals you'd like to work on and kind of have in hand at the end of the day. And like I said, check in with your group once a day to go over progress. Use Slack, use GatherTown, use Jitsi, use the resources you have to keep in touch, and just be flexible with it. So you may not meet your end of the week goal, and if that happens, that's okay. Adjust as necessary. This is kind of how hackathons go. Stuff comes up. Sometimes you don't meet all of your goals. Sometimes you meet way more than you thought you would have met. So just try and use this week as a time to have fun and make progress on these sorry, I'm losing my words, on these teams that you're working on for your projects. Okay, and then last thing I just want to kind of point out throughout the week, we really hope that you use this time to make your work as reproducible as possible. Like I said, there's going to be ReproNim helpers available throughout the Slack across project week. And I really hope you use this opportunity to kind of play with and learn these skills that you might not be as familiar with. And then on Friday, starting at noon Eastern Standard Time, we're going to start wrapping up the progress that you've made on your projects. So find a good place to pause in the progress that you've been making, and then work on creating a presentation kind of showing off what you were able to achieve during the week. And hopefully, we'd love to see you discuss future directions and make plans to keep collaborating with your teams. And then we'll end project week at 3 p.m. Eastern Standard Time, going over project presentations. It'll be in this room in GatherTown, and it'll be similar to how we do it today, where you all stand up on the podium and present what you were able to achieve across the week. So we're aiming for about 7-minute presentations on those. So these are all the projects that we have submitted for Project Week, and it's a lot, and we're really excited about all of them because they look super cool, and we hope that you're able to make a lot of progress on them. I can't wait to see what you're able to achieve by the end of the week. There are a lot of choices to choose from if you haven't already picked a project team. So please do pick one if you haven't already. We are requesting that there be at least 2 people per project. Please don't work on a project by yourself. But you can join more than one team if you want. And if you proposed a project but nobody joined your team, that's okay. You can still use today to try and get people to join your team. And also, if you have an amazing project idea that you weren't able to kind of put on the GitHub in time, that's okay. We're not going to stop you from pitching it today. Just send us a message and you can jump up on the stage right now and give your pitch. All right. So one last call. We do really hope that you use this week to kind of practice your reproducible science skills. We hope that you consider trying to containerize your work. We hope that you use GitHub for code versioning. We hope you try and explore with bidsifying your data or working with Bids app through your analyses and using DataLad and other tools for workflow management. If you have any questions about these tools, post them to the Project Week channel, because we will be active on there and trying to answer and help with any issues that come up. And again, we do recommend discussing authorship with your team members at this stage, just because it's good to discuss it at the beginning and throughout the process. And we do hope that some of these projects eventually turn into publications for you guys. If you run into any problems downloading ABCD data from the NDA, do take a look at the Project Week prep data exercises. They're helpful, and they're tutorials on basically how to download that data. So give those a look if you haven't already. And with that, I would love to welcome up our project team delegates to tell us about what your plans are for the week. Anyone who hasn't chosen a team yet, please use this opportunity to do so, and you can follow along at the projects here at the issues pages on our GitHub. And also message the people who are presenting now for each project if you want to join their team. Okay, so that was my mini welcome. Let me exit this screen share. Let's see if I stop sharing. There we go. Okay. So the first project that I want to welcome onto the screen here is a pre-adolescent imaging brain-based parcellation, which is issue 21 on the GitHub. So I'm gonna jump over here to this white podium. Anyone who is on this team or the person who's presenting, Helen, I already see you're here, great. If you could step up to the podium with the rest of us, and I guess Angie, Satra, Dave, let's give space for those other people by walking off the stage. I will just call people up when we're done. And if you have any questions for anybody giving these presentations, you can come up to the two podiums on either side of the stage, and you'll be broadcasted for the whole room. So Helen, thanks so much. And let's see what you guys are planning for the week.
Hi, everyone. Can you hear me?
Yes.
Okay, good. Well, my name is Helena Hopman, and I was inspired by Jessica Devlin's proposal about brain parcellation in pre-adolescents. Our team has currently 5 members, including Julio, Jessica, Grace, Dustin, and me. And the aim of our project is to create a functional brain parcellation from healthy pre-adolescent subjects using the ABC dataset. This parcellation scheme can hopefully be used as a reference for future studies. Our project is mainly inspired by Yeo and colleagues, a publication in 2011. They used a clustering algorithm and analyzed the stability of the clustering algorithm to identify the optimal number of clusters for the parcellation scheme. We will adjust their scripts, which were written in MATLAB. However, in the spirit of open science, we will perform the analysis in Octave. This is an open source alternative to MATLAB. During this week, there are several issues we need to solve. First, we need to examine the effect of different motion thresholds. Here, it's important to find a balance between having good quality data, but also being inclusive enough so that the parcellation scheme can be generalized to other samples. So therefore, we will examine several thresholds on a selected range of subjects. And after we define the motion threshold, we would determine the optimal number of clusters by analyzing the stability of the clustering algorithm, which is similar to what Yeo did. The tools we will use are GitHub, DataLad, and a container with Octave. And our long-term goals are that we would like to translate the code to Python, because we believe that that will benefit the neuroscience community. And second, we would like to compare baseline measurement with the follow-up to examine change over time. So that's our plan for this week.
Great.
Thank you.
Thank you so much, Helen. I think that's a great plan and a good scope for this week too. I'm really excited to see what this project gets at the end of the week. So if you could stop sharing your screen by pressing that little button on the bottom down there. And I invite up the representative from our second presentation here, Childhood Unpredictability and Reward Processing. Lillian, or is it Grace who's presenting? I think you need to stand right in behind the podium if you haven't. Yeah, now we can hear you. Perfect.
Okay, great. So can you now see my screen?
Yes, we can.
Okay. Hi, I'm Lillian, a grad student at University of Wisconsin-Madison, and our project will look at childhood unpredictability and reward processing. We have Mignon, Cassandra, Clara, and me on this team. So for the project week, our scope will be limited to first extract an unpredictability score from measures of childhood adversity. And then we're going to examine whether reward processing in a monetary incentive delay task is associated with childhood unpredictability. And importantly, we're going to use the tabulated baseline data for the neuroimaging analysis because we don't have time to start neuroimaging analysis from scratch, and we want to focus on one data wave at one time. We have 3 specific goals. So first, we're going to select and compute variables indicating unpredictability in each wave of the data. That is baseline, 1-year follow-up, and 2-year follow-up. And secondly, we're going to use a factor analysis to inspect the latent structures of unpredictability variables for each data wave, just to see if there's a set pattern across time. And then we're going to derive a composite score of unpredictability for the baseline data only. And with that score, we can go ahead to examine the relationship between childhood unpredictability and reward processing, both at the levels of brain and behavior in a MID task. So for our roadmap, if everything's in their best shape, we hope to complete the first and second goal, which is about our predictor on predictability, by Wednesday, so that we can start working on our third goal by Friday. But I totally understand how complicated data analysis can be. So the plan B is to finish the first goal by Wednesday and the second goal by Friday, if possible. And in terms of the expected tech support, so we would like some guidance on collaborative coding workflow, because we want our team members to help out each other as much as possible, but don't want to risk wiping out each other's work. So any best practices on that or guidance on that would be great. And secondly, in terms of the specific coding support, we would appreciate if anyone has Python or R scripts for dimension reduction, ROI neuroimaging data analysis, and cross-validation. So if you can share those with me, with us, we would really appreciate. Yeah, so I think that's our project proposal for this week.
Thank you so much, Lillian. That was very clear. And if anyone wants to join your team, please contact Lillian on the Slack. All right, so I'm going to invite up the next group here, which is the effects of race and socioeconomic status on mental health trajectories of adolescents and older children. So let's see, who is the representative for that team?
Hi, that's me.
Oh, that's Grace. Yes, hi, Grace.
Is my audio clear?
Yep. Okay. And you can share your screen by presenting or pushing the screen share button at the bottom of the page. Perfect.
Okay.
All right. Hi, everyone. My name is Grace Jacobs, and I'm representing our project team. So we're interested in looking at the effect of race and socioeconomic status on mental health trajectories of children from the ABCD study. So there are a number of studies that have found associations between socioeconomic status and brain structure, as well as SES and mental health, but not necessarily looked at these variables together. There's also evidence that these relationships can differ based off of race, and that brain changes may be related to accelerated pubertal development that can occur in response to childhood adversity. So for this project, we're interested in building on these findings by examining the relationship between race and socioeconomic status with mental health trajectories, and how these relationships may be mediated or moderated by brain structure and pubertal development. So far we've set up our Jupyter project folder and GitHub repository and started to meet to discuss kind of the details of the variables and delegating tasks. But definitely feel free to join our team. And by midweek, we're hoping to narrow down the details of exactly what we're going to include in the covariates and the structure of our analysis approach, which will likely be structural equation modeling. And by the end of the week, we're really hoping that we'll have some preliminary results, tables and figures, and a really clear action plan to divide up the remaining tasks between group members moving forward. So that's my whole presentation.
Awesome. Thank you so much, Grace. That sounds super interesting. I'm looking forward to seeing what you guys get at the end of the week. Let's see. I would like to invite our next representative up to the podium here. Functional connectivity in emotional working memory and anxiety. Hi, Morgan. I think you might be muted.
Am I in the right place?
Yes, you are in the right place. You want to share your screen with us?
Gonna try.
The button at the bottom of the window.
Oh no, it needs permissions. Okay, wait.
If you need to go set permissions, we could move on to the next team. Yeah. Okay. So we'll call you back up in just a second, Morgan. We are gonna call the next team now, though, which is typologies of temperament, identifying adolescent temperament profiles that predict substance use risk factors. I see Lauren coming up to the front. Hi, Lauren. Can you unmute yourself? Oh no, we can't hear you. Can you say something again? Oh, now we can hear you. Okay, great.
Okay, it's working now?
Yeah, it's working now.
Okay. Alrighty, so let me share my screen and I will get started. Okay, can you see my screen okay?
Yeah.
Awesome. Alrighty, so I'm Lauren. I am with the Typologies of Temperament group. And so I'll be briefly going over our project plan for the week. So just a quick reminder about some of our research questions. So first being that we want to look at the study-specific temperament dimensions and typologies within the ABCD cohort. And then second, we want to see if these typologies and profiles parallel prior work or offer a novel solution. And then we finally want to see if these temperamental profiles predict neurocognitive risk factors for psychopathology. So our specific goals are to implement open science principles. So by using DataLad and GitHub, examine the demographics and descriptives of our specific sample, and then take into account those nested variables such as site and family. We want to prep our input data, so confirm our indicator variables that'll be going into our latent profile analysis, and then also calculate the percentages of missing data in these variables as well and scaling them. And then we will define our latent profile analysis model fit criteria based on prior work. So looking at AIC, BIC, entropy, etc. And then finally identify the psychopathology risk measures for our regressions that we would like to look at. So we have specific project week milestones. So at the beginning of the week, getting our Hub directory and GitHub up and running, as well as getting a clean CSV file of the variables of interest. And then Tuesday, running our latent profile analysis for covariates, and then looking at maybe biological differences between males and females, and then address model fit criteria at that point. So midweek, hopefully interpret our latent profile analysis, clustering profiles, and run our logistic regressions. And then Thursday, compiling our results and figures, putting everything together for Friday to present our results. And so the technology and resources we will be using is our BIDS data standard, data lab, and GitHub. And that is the end of it for us.
Thank you so much, Lauren. I love how detailed your daily plans are. Fingers crossed that you get everything that you need by the end of the week.
I know, me too. Thank you.
Morgan, can you give me a thumbs up if your screen sharing is ready? Okay, why don't you come back up here then? All right, Morgan's going to talk about her project, Functional Connectivity in Emotional Working Memory and Anxiety.
I do believe it's working. So let's check.
It's working.
Yay, great. Okay, so here are the people on the project, and our project scope, we have some ambitious goals, but we're hoping to achieve them. Our overarching goal is to learn from each other and document everything so we can use this framework as for future analyses. But also we want to make sure that everyone understands everything with the whole project, because no one wants to have a presentation later down the line for a conference for work that you did and not be able to answer a question. So we want to make sure that everybody really understands everything that's going on, whether or not you helped with something. Our general question transitioned a little bit further from functional connectivity to neural activation. So what we're looking at is, how does neural activation during the emotional working memory task, the NBAC, relate to anxiety? So thus far, we have done quite a bit. We have examined potential measures of anxiety, looked at what the outputs and the derivatives are for the emotional NBAC task, and then find and read relevant literature. Still reading relevant literature. The next steps are to decide measures for anxiety. So do we want to use the KSADS or the CBCL? Decide on an analytic strategy. We're thinking something similar to O'Brien et al. 2020, but that's something that we'll mention a little bit later. Our specific goals on Monday, Monday, yeah. We're going to finalize our hypothesis and analytic strategy. That's our goal for today. We've already done quite a bit up until noon EST. And then tomorrow we're going to work on our task-based fMRI analyses. Wednesday, our goal is to finish our task-based fMRI analyses. Thursday, we're hopefully going to start the behavioral data. So in relation to anxiety, and then how these kind of relate to the NBAC task. So reaction time or accuracy maybe. And then Friday, hopefully we can make some figures and then present all of our findings thus far. Project milestones, I just mentioned kind of how midweek and end of week just a little bit more clearly, and potential areas for assistance. So like I mentioned that O'Brien et al., the advice on the statistical approach, we are toying with what would be the best way to do things. Some of our members in our team have more resting state experience, so it's not really task-based, and then I have more experience on setting up analyses, which has already kind of been done for all of these data, because it's, yeah. So we're kind of looking at what is the best option. That's what we're reading a lot. So if there's anything that we could do better, we're willing to hear it. Also, we want to ensure that reproducibility is up to par, and mostly for version control and containerization. And yeah, that's it.
Awesome. Thank you so much, Morgan. I think that's doable within a week. So fingers crossed for you guys. Good luck. All right. So let's invite our next group up, which is shared and distinct neural correlates of reading and attention. Hi, Marissa. I think your video and audio might be off. There you go.
All right, awesome.
Welcome.
Hello.
Want to share your screen? Perfect.
All right, can we all see that?
Yep.
Perfect. So we are working on the shared and distinct neural correlates of reading and attention. 1 second, if it wants to share. There we go. So just a recap of what this project is in general, we are going to be looking for correlations between resting state functional connectivity and both reading and attention scores to see if these are relatively distinct or overlap with each other. And this should help us understand why there's such a high comorbidity between reading and attention disorders. We hope to use the cognitive toolbox task for oral reading recognition and the flanker. And we do have some stretch goals in case this goes surprisingly well and we have time to work on more things, hopefully applying genetics and also seeing if there's a moderating effect of processing speed. And so for project week, since that is a lot, we hope to focus on reading and creating the proper pipeline, since this is all a fairly new area of research for us. And so we, thank you, Destin, for helping us out with this, but decided that the FSL pipeline might be our best option. And so we hope to use melodic and dual regression, and then end with FSL nets to do the connectivity matrix. We hope to do this in Octave, which, as was stated previously, is the open source like MATLAB alternative to help get over the paywall options. And so for midweek, we hope to obtain the proper resting state and reading data from approximately 15 to 20 participants. Like I said, since this is a new pipeline for us, we don't want to throw everybody in right away. And we hope also by midweek to have the first step, the ICA analyses complete. And by the end of the week, we hope to finish that pipeline in the small sample size and hopefully use it on the larger sample size. And again, some stretch goals that we have are to replicate that in another large sample from the ABCD data set, and also to apply this pipeline to the attention scores. And we do think we might need a possible assistance running the commands in Octave because we have not used this before. And again, also with DataLad, just because this is fairly new to us as well. So thank you.
Awesome. Oh, I'm muted. Awesome. Thank you so much, Marissa. All right. Let's see. Let's welcome our next group up, which is association between structural and connectivity characteristics in the language networks and the statistical learning networks and children's language abilities. GitHub issue number 15. Awesome. Thanks, Catherine. Hi there. There we go. We see it.
Okay. I'm sorry. I'm just trying to make sure it is easier to see.
No worries.
Okay. So we are studying the relationship between statistical learning and language skills in children from a brain connectivity perspective. And to do so, we will be looking at the correlation of statistical learning tracks implicated by adults with children's language skills and the features that correspond to high or low performance. We will also, as time allows, examine the correlation of functional connectivity of our statistical learning ROIs with the children's language skills and see how the correlated ROIs overlap with our tracks. And finally, we will compare these outcomes to the traditionally implicated language tracks to see whether or not they overlap between the two. So to do so, based on the regions of interest from adult statistical learning data and the diffuse intention imaging results, we will use DSI Studio to isolate the white matter tracks to correspond to the functional statistical learning language regions identified. We'll then create a pipeline on Jupyter Hub that will again use DSI Studio that will take the MRI results from baseline children that fit our exclusion and inclusion criteria and look first at what tracks correlate with the children's age match scores in the NIH's picture vocabulary and oral reading tasks, and B, how high performing versus low performing children on the language task differ in their structural connectivity. We will also look at our regions of interest and deep as time allows via brain space points and use that to look at which regions functionally correlate with the children's age max scores in the NIH's picture vocabulary and oral reading tasks. And then finally, we will compare our structural results to see how they overlap with the areas most strongly correlated by the functional results, along with whether or not they align with the tracks most commonly implicated in language processing. Our goal by Wednesday is to have everything established in terms of materials and pipelines we need and to begin running them. And then by Friday to have completed running the pipeline and to have a basic analysis of the results and how they overlap with one another and tracks in the literature completed. And so people are still welcome to join us. We will be meeting later today to discuss it. So if you don't have a group and you're interested in statistical learning and language, we'd love for you to hop on. Thank you.
Thanks so much, Catherine. All right. Okay, let's move on to our next group, which is, let's see, specificity of structural brain markers of psychopathology risk. Hi, Matt. Welcome.
Hi, is it on?
Yep.
Okay, let's see. You see the slideshow?
Yep.
Great. So as a reminder, our main project is just going to be focusing on how differential parental history of psychopathology, such as like depression or substance use, anxiety, affects neuroanatomical abnormalities and psychopathology risk in the youth. And this is building on a study that was already done in the ABCD data set by David Pagliaccio and colleagues that really focused on depression. And we're hoping to expand that to see if some of the effects are specific to depression or are related to comorbidity or just different other disorders as well. So we're specifically going to be looking at, does parental history of depression, anxiety, substance use, or alcohol use affect youth's cortical volume, cortical thickness, as well as psychopathology as measured by the case sets. We've developed a little bit of a project plan, trying to finalize what variables we want to be looking at. Our main variables are going to be the parental history variables, as well as some of the free CFR outputs for MRI and the parental report for case sets. Of course, we want to consider how to use ABCD-ReproNim. And I think how we're going to plan to incorporate that right now is try to build a pre-registration, which can really like finalize what variables you want to look at and like make some a priori decisions of how we want to finalize everything. And then that's kind of like a midweek goal right now, optimistically. But if we can get that done at some point during this week, I think it'd be a really useful document to kind of continue on to make sure this project is still like feasible in the future. Then like optimistic end week goal would be to use like a pre-registered analysis approach to run a preliminary model and begin looking at least some few regions of interest to see what we get. Elena has been working on some data lad, but I think we could probably use some help from the ReproNim team with using data lad and get version control to make sure everything is reproducible. And we also are thinking about how we can make sure our model is not too complex so we can get some like real interpretable results. That is it.
Awesome. Thank you so much, Matt. All right. Let's welcome our next group up here, which is Neural Correlates of Pain in Youth, a preliminary investigation to inform future discovery of future biomarkers. Hi, Moab.
Hi, can you see me?
Yes, I can.
All right, let me
share your screen
share. Okay, 1 second. Share this. And where's my window? I'm sorry.
Oh no. Awesome, there we go.
Can you see that?
Yeah.
All right.
It's so not Inception.
So we'd like to welcome Ranjita. She joined us. So Sarah, myself, and Ranjita are working on this project. Again, this project started by the fact that ABCD finally started to collect data about pain, and we are interested in pain and what would predict people from being painful or non-painful using functional connectivity. So this is the main goal. But we also want to know if there are risk and resilience factors that interact with developing pain, or how they affect the functional connectivity, or how they affect your brain activity. So it's all going to be based on resting state functional connectivity analysis. So the scope of the week is we would like to finalize the selection of the variables that we want to analyze. And I'm referring to variables that we are going to include in our models beyond the functional connectivity variables, such as related to cognition, related to psychiatric conditions, physical activity, and so on. We would like to also visualize these variables to understand them better. And also we are going to start working on mixed effect models where we are using pain and other variables to describe variability in functional connectivity. And by the end, we would like to outline and draft a plan for machine learning. So we know that inverse inference is not the best thing to do. So we would like to be able to say, here is what we know about that participant from brain activity and other collected data about them. Can this combination of data predict if they are painful or not? So the plan is we're going to analyze and select the variables that we want to explore. We can decide on visualization using Python and GitHub. At least Sarah and I, we don't have experience using Python. We use other coding programs, so Python is new to us. And we're also going to use R and SAS and MATLAB to analyze our mixed effect models, and we're going to compare our codes as we move between the three different languages. So by midweek, we'd like to have the variables selected and visualized and understood. We might need a feedback on what variables that we should include. We have a very good idea about the variables, but we also would like some feedback maybe from our TA, Christina, since we have been working with her for a while. By the end of the week, we will run preliminary mixed effect models by Friday. We'll also have a draft for our machine learning. And we might need evidence with whether our code of mixed effect models and machine learning is on track. So one thing that we know that we are not going to do this week is to download the resting state fMRI data and create connectomes for each person using the FOMO, but we would love to use the FOMO in the future. So we need help on understanding that process. So what we are replacing the FOMO with is Gordon connectivity tabulated data, and we will see whether this, lower number of brain regions might still predict pain or not. So it could be helpful, and we don't need to go to the FOMO. But if it is not, we would know by that time that we need to have many more regions of the brain and do IC analysis on them before feeding them into machine learning. So that's all, and we would like to thank everybody that made this course possible for us. Thank you.
Thanks, Bob. All right, well, thank you for your presentation. I hope that if anyone's interested in your group, they join it too. Let's move on to the next group here, which is development of amygdala functional connectivity, GitHub issue number 19. This is a very interesting screen that is being shared right now.
What is that? All right. There you go.
Thanks. All right, Ali, are you with the neural correlates of pain? I'm sorry, the development of amygdala functional connectivity?
Yes.
Awesome, cool. We can see your screen.
You just see the slides?
Yes.
Okay, perfect. So yeah, my name is Allie. Just a reminder, this is development of amygdala functional connectivity. Since my proposal last week, I've had Megan join my team, so it's super exciting. Our overall goal is just to explore the relationship between amygdala functional connectivity and measures of early life adversity. And so our plan for the week starts with designing a construct of early life adversity using the assessments sort of collected from this ABCD cohort. And then we're going to validate it with factor analysis to make sure we're sort of capturing what we're interested in. We're then hoping to leverage the tabulated correlations between the amygdala and the frontoparietal network. So this is a little bit different than us sort of conducting our own analysis from scratch, since these are already sort of calculated from the resting state data. And I've done some research in the frontoparietal network. It does seem to get at what we're interested in, but if anyone has like thoughts about using this sort of network approach, or would rather see us, just work with something more specific, like just the medial prefrontal cortex, please let us know, since we are open to running our own, but we thought to maybe reduce the computational load for the week since there are only 2 of us. And then after we have that data, we'll analyze the relationship between individual differences in functional connectivity and our early life adversity variable that we create. And so our midweek goal is to have that measure of adversity finalized, validated, and calculated for our participants, and then have the appropriate resting state data sort of figured out and downloaded or extracted in some way, with the understanding that if we are going to run our own analysis, that'll take us a little bit longer. By Friday, we're hoping to complete some preliminary analyses and have some visualizations for you, hopefully, and also kind of discuss the future directions. In terms of resources, we'll be using Python and R. I don't know why there's a G there. It should just be R. And then some, reproducible practices like GitHub, Data Lab, etc. In terms of collaboration, we're already talking a little bit with Anna Vanucci's group about our childhood adversity variables, since they're also looking at that. And we'll probably reach out throughout the week to the group about ReproNim best practices and sort of our final statistical procedures. But yeah, that's it. We're really excited for the week.
Awesome. I love hearing intergroup collaborations, and I love hearing the goals about using reproducible tools. So thank you so much, Allie. All right, let's move on to our second to last presentation actually here, which is Early Life Adversity Dimensions, Functional Connectivity, and Change in Child Behavior, issue number 17. Anna, if you want to step a little bit down. Yeah, perfect.
Okay, I'm at the right spot?
You are at the right spot. We can see and hear you.
Thank you. Yeah, we've been talking, our group has been talking with the 2 other groups looking at early life adversity, and I think we're trying to do a lot of knowledge sharing there. That's and awesome. Potential harmonization of efforts and knowledge that we're doing.
That's great.
Yeah, it's really nice to hear other people's thoughts and just kind of meet other young individuals in this field. I'm only in my 2nd year of grad school, so so it's I don't know that many people. So so our project is, we have quite a team and quite an agenda. Our project has been split into 3 sub-projects that will each be their own paper. Because each one is kind of a big question. But I came into this course with a strong idea that I wanted to look at the relationship between early life adversity dimensions that were data-driven, whole brain functional connectivity, and change and internalizing and externalizing behaviors in this sample. We have a large project team, including an awesome TA liaison, Adam Richie Halford, who we already have several meetings set up with, and met with him last week, and he's been very engaged, and it's awesome. He's interested in things. So our project week goals, and I just want to split things up into the 3 projects. So the first project is to complete, well, involves several team members, but not all of them. We're basically subsetting ourselves into different teams as we complete these projects. So our first goal is to identify the factor structure of early life adversity dimensions in the data. Prior to project week, we've been meeting really since November, and so we've done all of our variable selection. We've created a detailed analytic plan, as well as have completed looking for outliers and missing data and making our exclusions and things like that. So And with regard to project 2, we are really, really excited about the opportunity to learn and teach ourselves with the assistance of Adam and others here, how to do connectome-based predictive modeling, because I think this is a wonderful opportunity to try to really do the gold standard of predictive modeling, where we have like our training set, our test set, and then a final validation set after all 3 pipelines have been finalized. And we would like to, for this week, try to get a clean pipeline running for at least one of the adversity dimensions, and if time, more. And yeah, so that's really our goal for project 2. And then project 3, since things kind of have to occur in a stepped order, we have tentative goals for project 3, potentially get an initial causal mediation pipeline generated with like placeholder variables of like the early life adversity dimensions, and then maybe like our initial run of experience sensitive connectomes. And similarly for all of these, we've completed our variable selection. We extracted the decan derivatives for the ROI time series. I'm really happy that those were available. And we've calculated our functional connectivity matrices and are currently just making, like standardizing them, like doing Fisher's R to Z transformations, which we've like done this morning. So in terms of our milestones, our goal is to, we just about finished up data cleaning actually this morning for the factor analysis, but today in general for our group, it's really just a data cleaning day and creating our train test validation sets. And for the factor analysis, we are coming in at a little bit of an advantage because we have existing scripts that we're going to be just adapting for this purpose. And we plan to run a bagging enhanced factor analysis. Day 2 will really be, our hope is to get some initial variables from the factor analysis to use as test holders or placeholders to generate the CPM pipeline. And our big goals for day 2 is really to be optimizing parameters for the factor analysis and begin testing. On our training set, we plan to do use cross-validation to test a number of parameters to see what's going to be optimal in this particular data set. We're using that based on, we're kind of taking the approach in the from the Dottie et al. Paper that did benchmarking of connectome-based predictive modeling. Wednesday, we plan to test and launch our full bagged factor analysis script and begin all of our plotting and really interpretation of those factors. On Wednesday, we plan to try to finalize what our ultimate CPM model is going to be, and really run those scripts, final scripts in the training set. And for the third project, it'll be kind of beginning to assess the relationships among these variables as we get them. So for our midweek goals, we plan to we would like to launch our final factor analysis script. We would like to finalize our CPM parameters. And then for question 3, assess the relationship among the variables, just in a really informal way. For Thursday, we'd like to just do some kind of validity testing with the dimensions, plot the results, and we want to launch our optimized model for the CPM model for the ELA dimensions, as well as do some plotting. And then for question 3, begin the mediation model in the training set. And so on Thursday, we hope to, for the connectome-based predictive modeling, do that initial test set, get the initial test set prediction performance, and basically all of Friday we'll be compiling slides and taking care of little things. So we hope to have a full factor analysis pipeline complete, a pretty clean CPM pipeline for at least one dimension, and hopefully just run an initial path analysis. Our resources will be using R, Python, potentially Connectome Workbench, potentially MATLAB, although we're trying to stay away from MATLAB and just work in Python. For reproducibility, we'll be using the bid standards, GitHub. We are flirting with Data Lab, but are kind of scared of it. So if anyone could help us on that level, that would be really helpful. And we also would really like to containerize our analyses and the environment that we're working with. So that would be helpful. And then I think we'll need a lot of help with CPM. But Adam has, like I said, we've already had some meetings. So that's our wrap up. And thanks so much. I'm really excited for this week.
I am too, Anna. Thank you so much for your presentation. I love the different levels of like detail that you're going into the project. And definitely, for sure, there are many people here that will be able to help you with using Data Lab and containerizing things. Just post it to the Project Week channel and you will receive some help with that. All right, so let's see. We have one more group with the presentation. I see Claire coming up here. Using multivariate statistical techniques to investigate the role of brain volume and nutrient density on psychopathology.
Hi, yeah, can you guys hear me?
Yes.
Okay, good. Let me share my screen.
Oh, before you go into your presentation, go ahead, share your screen, Claire, but I just was noticing that apparently some people are having some issues where potentially the screen sharing is either just a black screen or you can see it. If you like, I don't know, maybe GatherTown might be a little bit buggy on this for the, I don't know, hopefully it's working for most people, but if it is buggy for you, just log out and then log back in and it should work okay, or like you can even move around and maybe that'll help it too. But yeah, okay, thanks, go for it, Claire.
Okay, so this is just a quick overview of what we're planning on doing this week. So our scope, like we talked about, so it's my team, Carlos and Rachel, and we'll be trying to figure out whether brain volumes interact with the RSI measurements that are pre-tabulated in the ABCD data and whether that affects, that explains any adolescent psychopathology. We're planning on sticking with the baseline data right now and using it while using the FAIR principles. So our main goal is to utilize everything we've learned over these past few weeks and use the FAIR principles correctly and learn how to use them collaboratively between people, between OSs, and how do we then transfer everything so everybody can download it and reproduce our data. So the technologies we're trying to use are the Jupyter Hub, and we're also just going to need a lot of help trying to figure out how to containerize, how we're going to track all of our code edits, all while collaborating. And this is just a brief overview of our analysis plan. So then briefly, our next thing is our midweek goals. We want to make sure we can pre-register our plan. We also then want to containerize. We're going to clean all the data, view our descriptives of all the data, and then hopefully run the PLS, which we already have code for, that Carlos has already done before. And I forgot to add Rachel's name here. Sorry, Rachel. And then our end of week goals, we're really trying to then visualize our data and hopefully we can see what our results look like. And then make sure our analysis plan is actually reproducible. So hopefully it will be able to all be downloaded correctly and somebody else can do it if they have access to ABCD. And then we're also thinking about potentially transferring the PLS code that Carlos has in MATLAB to something that is more open sourced. And then, so what we'll need assistance with is just all the ReproNim tools. We're just really trying to utilize them. So if we're going to use Git or Data Lab for like tracking our changes, and then also containerizing, like how to use that when you're collaborating. Do you all use the same container to make sure that you're using the same versions? And then setting up a Jupyter Hub so we can all use the same code and data. But that's just a brief overview of what we're planning on doing.
Thank you so much, Claire. I love your goals of making everything in line with FAIR principles. And this is the perfect week to get help
Yeah.
on doing that. So thank you
That's
so much.
very new to us, so we're trying.
Yay, that is what this project week is for. Okay, so that was all of our presentations. Thank you so much, everybody, for presenting and writing out a plan of what you plan to do this week. We budgeted a little bit more time than we needed. So the next thing on our schedule here was meeting up with your project team at 2 p.m. You can feel free to meet up with them now, or take a little bit break, grab some late lunch if you haven't already eaten lunch over wherever you are. And I just wanted to, before we sign off from this beginning kickoff portion of things, remember that all of the ABCD-ReproNim organizers, Angie Laird, Dave Kennedy, Satra Ghosh, Jibby Pauline, myself, will all be lurking around the Slack channel. So if you have any questions, just tag one of us and we'll see it. And we can help with the kind of things like that you were talking about, Claire, or that other teams were talking about that are ReproNim related. Like if you're flirting with Data Lad and you are having some problems, but you really want to learn how to do it, post a question, tag us, and we'll be there to help. All right, well, with that, thank you so much, everybody. I am so excited to see what comes out of this week. I'll do a little dance because I'm so excited. All right, cool. Well, thanks, guys. Let's wrap up this intro kind of portion and go forth and project. Touch base with your teams. Remember, touch base with your teams at least once a day. And good luck. We'll talk to you on the Slack. Thanks, everybody.